Hi everyone. So today I'll be discussing and showing how to perform whole genome amplification using the Equiphi polymerase DNA kits. So with the Equiphi DNA polymerase kits, you can amplify your a whole genome using either your DNA as your starting material or complementary DNA as your starting material. So in this case, I'll be using the complementary DNA as the starting material. The complementary DNA as your starting material, first thing you need to do is to purify the cDNA to get rid of excess salt and your primers used in the synthesis of the complementary DNA. And to do that, I'm going to use 80% ethanol and magnetic beads. So first thing I'm going to do is I'm going to wear my gloves, then clean my workspace. I'm going to start with cDNA. So this is the cDNA I synthesized from the previous experiment which I showed you guys in the previous video. So to start this experiment or this protocol using the Equiphi DNA polymerase to amplify the whole genome from complementary DNA, first thing I'm going to do is to purify my cDNA. And to do that, I'm going to use magnetic beads and 80% ethanol. So, so the total volume of what I have in my PCR tubes, my starting material, which is the cDNA, is 20 microliters. And the magnetic beads, it's usually added or used in the ratio of 1:1 in this project or for this protocol. So I'm going to add 20 microliters of my magnetic beads to each tubes. So before that, I'm going to shake vigorously to suspend the magnetic beads. Then I'm going to add 20 microliter of my magnetic beads into each tube. So I'm going to use my P100 and I'm going to add 20 microliters of magnetic beads to each tube. So while opening the tube, I have to be very careful as not touch the rim. So I use my nails to hold the tip of the PCR tubes. So this is to avoid contamination. And I'm going to add 20 microliter of my magnetic beads to each tube. So to save tips, I try as much as possible not to make in contact with the PCR tube. So when I'm done, next thing I'm going to do is to flick the tube and centrifuge. So I'm going to allow to sit for 2 to 5 minutes. So after 5 minutes, I'll place the tubes on the magnetic rack. So you can see like the clear spinetons forming already. So when it's done, I'm going to pipette off those supernatants and discard. So using the P100, I'm going to pipette off the supernatant and discard without disturbing the pellets. So next thing I'm going to do is, using the P200, I'm going to wash with 200 microliter of ethanol. So this is my freshly prepared ethanol. So I'm going to wash with 200 microliter of ethanol. So during the first wash, I'm going to vortex the tube, but in this case I'll flick the tube because I prefer flicking, and centrifuge. So when I'm done, I'm going to place on the magnetic rack. I'm going to pipette off the ethanol and discard. So I'm going to wash the second time with 200 microliter of ethanol. So next thing is, I'm going to pipette off the ethanol without disturbing the pellets. So next step, I'm going to centrifuge to take off the remaining residual ethanol. And I'll place it back on the magnetic rack. So using the P20, I'll take off any residual ethanol. So after taking off the ethanol, next thing I'm going to do is to dilute with 10 microliter of nucleus-free water. So I'm going to place this on a PCR rack, and I'm going to dilute with 10 microliter of nucleus-free water. So using my P20, I'm going to dilute with 10 microliter. So I'm going to flick or vortex and centrifuge. After that, I'm going to allow to sit for 5 minutes at room temperature. So after 5 minutes, I'm going to place on the magnetic rack to dilute my nucleic acid, or my cDNA in this case. Always remember that this purification step is crucial if your starting material for whole genome amplification is complementary DNA. If it's nucleic acid, if it's DNA, you don't need to perform this step before using the Equiphi polymerase DNA kit. Or if it's a synthesized DNA, which was done using another kit, you need to perform this step before using the Equiphi DNA polymerase amplification kit. So I'm going to transfer my cDNA, my purified cDNA, into a new tube. So I'm going to number the two, 1 to 10, and my control. So using the P20, I'm going to dilute my CD. You should be careful not to disturb the pellets. So after dilution, next thing I'm going to do is to go ahead and perform the whole genome amplification using the Equiphi polymerase DNA kits. Next thing I'm going to do is to perform a master mix. So I'm going to perform a master mix of 12 samples. I have 11 samples, 10 original samples that I had from the beginning, from the previous video, and a sample control from the previous video as well, the protocol that I used for the synthesis of complementary DNA. I had a control. These controls are necessary to track in case if there is any contamination in your analysis or sample. You could always trace back to the roots where it's possible this occurred or happened. So now I have 11 samples in total, so plus a negative control that I'm going to add for this Equiphi DNA polymerase amplification kits. So I'm going to make a master mix for this sample, because if you can see, I'm working with small volumes here. So you can see 0.2, so I'm working with small volumes as well. So I'm going to make a master mix. So this master mix is to help me be in control of the whole process. So for the master mix, I need 26 microliter of Equiphi DNA buffer. So I need 26 microliters. I need 2.6 using the P10. I'm going to take 2.6 microliter of DTT. So next, I'm going to take 26 I'm going to take 13 microliter of exo-resistant random primer and 13 microliter of exo-resistant oligo-DT primer. So next, I'm going to take 26 microliter of DNTP mix using the P100. And I'm going to take 89.7 microliter of nucleus-free water. So I'm going to take 97 microliter of nucleus-free water. So my nucleus-free water, I'm going to take 97 microliter. So I'm going to mix either by flicking or pipetting up and down. So when I'm done, I'm going to centrifuge. Since I'll be adding 4 microliter of my DNA template, it means I'm going to add 14 microliter of my master mix in each tube for my samples. Always remember to label your tubes. So in each tube, I'm going to add 14 microliter of my master mix. So using the P20, I'm going to add 14 microliter of my master mix. So after adding 14 microliter of my master mix, next thing I'm going to do is add 4 microliter of each of my sample to the reaction mix. So using the P10, I'm going to add 4 microliter. So when I add my cDNA to the master mix, I'm going to pipette up and down 2 to 3 times. So after that, next thing I'm going to do is to set up a cycling protocol using the pharmacycler. So I'm going to denature my sample, my cDNA first. Before adding the polymerase, I'm going to denature my PCR, my cDNA. So I'm going to do that at 95 degrees Celsius for 3 minutes. So after 3 minutes, I'm going to stop the reaction and take my samples, then centrifuge. Then I'm going to add 1 microliter of Equiphi DNA polymerase. So I'm going to add 1 microliter of my Equiphi DNA polymerase to all reaction. So I'm going to mix by pipetting up and down 2 to 3 times. So after adding the Equiphi DNA polymerase, I'm going to add 1 microliter of pyrophosphate, inorganic pyrophosphate. So I'm going to add 1 microliter of inorganic pyrophosphatase to each reaction. So I'm going to mix by pipetting 2 to 3 times. After adding the pyrophosphatase, next thing I'm going to do is to set up the further PCR reaction. So I'm going to incubate at 45 degrees Celsius for 2 hours and then 65 degrees Celsius for 10 minutes. So the total time for the PCR reaction takes 2 hours 10 minutes. So after 2 hours 10 minutes, I can stop the PCR reaction for other subsequent reactions. And to perform the Oxford nanopore sequencing using either the flow cell or the mini ion, I'll first purify this Equiphi DNA PCR product using the purification step that I showed in the beginning of this video. Then I'm going to treat it with endonuclease, which is what I'm going to show you guys in the next video. So for today, I'm going to save the Equiphi DNA, the PCR product amplified using the Equiphi DNA polymerase for the next subsequent run and reaction.